Hello, ladies and gentlemen. This webinar on modern histopathology was recorded live on 12 March 2019. My name is Emmanuel Loeb. I am the CEO of Patho-Logica. I wish you a pleasant and an informative webinar. I will start with a short introduction. This introduction will be in the preclinical trials, the role of pathology in the safety and efficacy roles that the pathologists fulfill in this context. Then we will talk about bias problems among pathologists. And further, I will give some study examples to show the variety of work we perform. Then I will talk about a very important tool, and this is the MORF3 with image analysis. And I will finalize with a short summary. So I called it from supportive to dominant. You have to realize that a pathologist working in the industry with the development of drugs, we have actually two fields. It's the safety. The safety is also called toxicological pathology. And this is a quite traditional field in our profession. Their researchers is usually very interested in selection of the appropriate dose levels of the new drug and the evaluation of possible side effects, and all the work is done in a global and broader toxicological assessment, which pathology is part of it. Then there is the efficacy studies, and there this question arises: does the drugs have a significant activity? And many years this was pathology was supportive in this field. That means that there were other tools that could compare the efficacy of new developed drug compared to a licensed drug. So here we are comparing groups of animals with each other, and accurate work is very important in order to achieve some and to show some difference. We should also set up endpoints that we can quantitate. The histological features have to be quantitated and evaluated, and I will come up to this on this point later on. In this work, we also try to expose mechanism actions of the drug using our histological tools, and sometimes we all have unpredictable effects of the drugs that can be seen in the histology. This is a global graph I selected. Here you can see the impact of histology on preclinical studies. What we see here is actually 50 individual mutant mouse that you can see as different models. In green, you can see the clinical phenotypic hits of each model, and in red, you can see the histological hits. So what we achieved, or what the writer of this paper achieved, and so 60% of the cases there was at least one clinical abnormality. from this 60%, 63%, so from the 30 cases, histology contains an added information, and the last one, 7 cases, showed a significant morphological abnormalities not predicted by the clinical screening. So this graph shows us actually the possible potential of the contribution of histology to the preclinical trial. Okay, so now we dive in the bias problem among pathologists. We talked now and we showed you I showed you the safety and the efficacy. In order for the efficacy to be stronger and more reliable, there are some bias problems to solve out, and I will go inside the next sheet. So, as a definition, a bias can cause a systematic error in the result of the study. Bias can occur due to the design, performance, analysis, or reporting of the experimental data. For example, in the design phase, number of animals is essential for in the context of bias and the choice of the appropriate assay parameters. So actually, I can add to that a very solid protocol with a hypothesis can contribute to the process and to avoid bias. The performance, different pathologists work differently. You can work double-blinded, and you can know in all the groups, the treated group. We believe in Patho-Logica that double-blinded studies are better because you always, when you know all the treated groups, you always can do some errors and mistakes. The analysis itself is also a kind of choice. For decades, we used to work with a semi-quantitative manner. That means that we and score our parameters from 0 to 5. These days, and generally, you can also perform quantitative analysis using advanced tools, and this is a major change that has been more and more seen in efficacy studies. And of course, the reporting, the usage of statistical tools, and seeing that no errors during the reporting are taking place. So the bias problem has been guarded and known for histopathology for many years. The reason for it are, first of all, the usage of biological materials, in this case animals and models, and this can always be a source of bias. But the more important one is the human component. Here we see different pathologists with different sensitivities, and that's why a peer review among pathologists is very, very accepted tool to try to avoid the differences between pathologists. In recent years, and I can see it in meetings of pathologists, this problem is more discussed and pathologists have become more aware of this problem. This led to a reduction of bias studies by using an image analysis. We'll talk about image analysis in a moment. I'm trying to manipulate something, but it's not very easy, so I leave it like this. The histopathology and efficacy studies has been increased compared to the past. Why? Because of the power of the reliable histological data. So what we see is the known bias problem among pathologists is solving down by the awareness of the pathologist and by the usage of image analysis tools. The relevance of morphological for your product development. Here I mean that histology is giving us a visual morphological change, which no other tool can give us. Once it's more reliable, we can use it, and then more and more preclinical trials will use us as a #1 tool. Okay, so still in the context of bias, in safety studies, we have no tolerance for positive findings. We are working and are getting drugs in different concentrations, and actually, because of that, the risk of bias is quite low. But the situation changes in efficacy studies. In efficacy studies, we have an animal model, we have complex models, difficult models, we have more bioanalysis on each animal, and we are aiming towards the patient with our endpoints, and we have in the of our minds that has been done before the preclinical trial. This is more complex, and we will see few study examples to show the complexity and the variety of different models and how we deal with it as pathologists. So now I go to the next. The next part will be some models. In this particular one, you see a very it's a very simple example of this. So you can see here above the very thick epidermis of the miquimod-induced animal, and you can see below animals that has been giving dexamethasone, and these are two poles actually. These are two controls groups of two extremes. You should imagine that treated groups are falling in between these two groups. So the quantification of changes between the accuracy is really needed in order to see any differences between those groups. Another nice example is emphysema. Above you can see a totally normal lung tissue, and below you can see an affected lung with a rupture of alveoli, and you can see a lot of air is trapped in the alveoli, and these are again two poles, two extreme control groups, and our mission or challenge is to try and give each treatment group a number or a figure. In this particular model, the responsibility of the pathologist is to select the right representative areas to quantify, because in this slide, for example, you have totally normal lungs tissues. So it is our responsibility to select the right ones and to send it to the image analyzer for further analysis. Next model is a ALS model. You can see an SOD1 animal. Usually in ALS, we are interested in cell count. Here you can see above a spinal cord, left, right, and you can see some positive cells. This is for us very easy to quantify and just to count them. But once you want to see the GFAP and the EBA1 for astrocyte or microglia, then it becomes much more complex, and you need the automized counter eye of the image analyzer system. Another model we do a lot in Patho-Logica is the hind limb ischemia model for angiogenesis. This is the old world analysis as we used to do it before. You can see here, you can see the muscle tissue of the treated leg and the normal leg. You can see the atrophy, and below you can see the immunohistochemistry of the Fezzel's markers. And this is how we used to quantify those samples in the past. You see it's complex. It depends on if you can have here some artifacts above. And these days we are doing it this way. So here you see very nice free-floating sections. You see all the dots here that are presenting active new Fezzel formation, and it's very easy and nice for the system to quantify these pictures compared to the former one. We are facing here now also the goods of good technological abilities and also the image analysis that makes our work much more reliable. So you understood now that to avoid bias, we are using some external tools, and one of these tools is the morphometry or the image analysis. And I will invest you my time now to explain how this works. How do you work with it? So first of all, you need, for the image analysis, you need a professional, skilled personnel. This is somebody who knows the system, who knows what he's dealing with, and know how to extract a number from each logical picture that you give it. Once we have a project, we define, the pathologist define to the image analyzer the structure for analysis. This could be cells, it could be fibrotic tissues, areas, height of epithelium, like we saw in the first examples. It can be almost anything in that might function as an endpoint. Then there are fewer ways to analyze. You can do the whole slide analysis, scan the slide. We don't like that. There are reasons why we don't do it. We usually select the representative fields. This could be 5 fields, 3 fields, and then it depends on the complexity of the tissue, it depends on many factors, and we send the pictures to the image analyzer, and then we see the results. The results are usually confirmed by the pathologist. Very often we see one animal or two animals that have bizarre numbers. So these animals are confirmed back to the tissue. It's very important because such an animal can destroy the whole group with this number. And the data, the raw data, is usually given to the researcher. They are very keen on the raw data, but we include everything in our documentation report, the pathological report, including the number of fields, the magnification, and all the methods of the image analyzer. Image analyzer, good image analyzer can make wonderful things. Here, just a very simple H&E slide. And when we wanted to count the nuclei, image analyzer gave us some background changes by manipulations of the picture without avoiding any details. But in contrary, you see the very, very extent in red, and this is very easy for the machines to okay, this is how it goes. Usually we use the Image Pro Plus program, but it's more advanced. Our image analyzer added more facets to this program, and actually the 34 of pick dextran. On the right, you give the definition to the program. Every red dot, for example, is a positive result. And here beneath, sorry you can't see it, you get a figure. These figures are analyzed to later on to an Excel sheet, and these are the raw data. This is of the image analysis. So to summarize our talk, reduction in histopathological bias has led to increased use of histopathology in efficacy studies. This is something can stand behind. We have much more demand as pathologists in efficacy these days. Technological advances have supported this process. We saw it also the pre-flow detections in the context of the image analysis. Pathologists' awareness of the bias risk is critical for the study success. Examples of several studies show the variety of parameters that the pathologist needs to take into consideration for efficacy studies. The image analysis is the most important tool to avoid bias and to aim for a reliable quantitative result. The position of the histopathology in efficacy studies has been changing, becoming a predominant methodology in preclinical. So now I'm done with my presentation. I will be happy to answer questions. You can approach me via our website of Patho-Logica.com, and I will be very delighted to answer questions for this presentation. So thank you very, very much for attending this session, and bye-bye.